Okay, this week we're going to study methods of disease control and detection. Although you have a lot to read and to review videos, I thought I'd give you sort of some background on certain aspects, but you are expected to read as well as follow the lectures and the videos and the articles provided to prepare for the quizzes this week and eventually exams. Well, let's get started. Now for some terminology. Classic definition of an outbreak is the occurrence of more cases of disease than expected in a given area or population. But you have to take in some additional considerations. Now these include the following. Is there clustering of cases in the same area and place or among individuals with similar demographics and disease and risk factors? This is where we come up to a term called a cluster. A cluster of disease may have a common cause or may be due to unrelated reasons. Now, to give you something of a relationship here when it comes to the term cluster, in June of 1981, the very first cluster, which turned out to be the onset of AIDS, was reported by doctors in the journal Morbidity and Mortality Weekly Review. And they said, we have this cluster, this unusual number of gay men who are showing up with these unusual symptoms of extremely rare diseases. And they're in the San Francisco area. And what you basically are seeing was the beginning of individuals having the latter stages of HIV, where they started showing up with being immunosuppressed. They started seeing the opportunistic infections. Okay. And by about 84, 85, it was starting to be well understood that all of this started with a viral disease, the human immunodeficiency virus. Now, related to the previous note, there are some clear epidemiological links tying individuals together. There are characteristics, often across person, place, and time, that may connect two or more cases. Also, are viruses or bacteria related to each other and thus due to the same chain of transmission? Whole genome sequencing, WGS, which we'll talk a little bit about more later on, is one high-resolution approach to examine a large fraction of a pathogen's genome for its overall patterns and diversity. This may help to confirm epidemiological links or vice versa. And this is very important today. We have the molecular tools to understand certain organisms. If you take just E. coli, and what you do is, sorry, I had to pass a yawn there. You take E. coli, there's so many different versions of them. Some of them are very dangerous. Others are just used as simple laboratory experimental microbes. So they're sort of like the lab rats of the microbiology. So you have to make this determination. What is this? What does it seem like? But then can we identify perhaps certain new genes that have been inserted into it that cause disease, etc.? Also, does transmission continue despite disease control efforts. This is very important because it may be that the efforts are insufficient to control the disease. If transmission continues in a steady, exponential manner, personnel and infrastructure might be stretched thin, necessitating increased urgency or additional resources. Sometimes you'll see this happen in certain countries, in certain cultures, particularly in developing countries where they don't have even enough boots and overalls and gloves to take care of gloving the individuals up, to provide care for the individuals who are suffering from Ebola. And this happens, unfortunately. What is the public health importance of the outbreak? In other words, beyond a lot of new cases, is the disease severe? You have the public health importance. If you're getting lost there, by the way, one way to look at this is it a cold? Well, you feel bad for a few days and then you get back up. Or is it something like COVID or Ebola or SARS, where there's a lot more, the diseases are much more severe and can lead to a great number of deaths? And that brings us up to one of the other little points. Are a lot of people dying? Are people unable to go to work or to school? Now, as we continue terminology, what I'm trying to do is help you to get used to some of the terms that exist out there. When we talk about epidemiological links, this is a characteristic that links two cases, such as, for example, close contact between two people, or a common exposure, such as spending time at the same place together. It is something to keep in mind when you have situations where you have a lot of people confined in some tight quarters, such as classroom, military barracks, dormitories. And are these situations that are conducive to a rapid spread of the disease up and down the hallways, shall we say, or up and down the area? Now, whole genome sequencing, this is a laboratory process that's used to determine a complete DNA or RNA sequence. Now, remember, RNA is necessary in part, if you're wondering, because a lot of viruses have their genomic information as RNA, not DNA. Now, this can be useful in tracking disease outbreaks. What's a case definition? This is a set of criteria used to classify individuals as having the health condition being assessed. They may include only the symptomology or may include laboratory confirmation. Remember symptomology, they've got a fever or a rash or any of a number of other observable conditions, but they may also have laboratory confirmations. Then we have active and passive surveillance. Active surveillance, this is a systematic identification and location of cases of disease, including screening, diagnosis, and treatment. Now, passive surveillance is the detection of cases among those seeking medical care who are both aware of their own symptoms and have access to healthcare facilities. This is one of the problems that we have today, is that some individuals don't always have free and easy health care. Now, some of you might be even wondering, well, what about, in the United States? No way, and it happens. You have the Midwest, you have the Rocky Mountain area. And if you look at the total number of doctors, let alone health clinics, it's certainly not as much as, let's say, if you went to East Coast states or the West Coast. Okay? So we move on from here. Now, when we talk about the methods of disease control and detection, there's a process model for early outbreak detection. I want you to note that the model shows the role of the epidemiologist in the left column and the decisions that are based on the investigation in the outputs in the right. So in the early stages, up in here, outbreak in particular, epidemiologists will play key roles of collecting and managing and analyzing data to ascertain whether a signal is truly indicative of an outbreak and a true increase in disease is occurring. We could talk about measles. We could talk about rubella or polio. And if you sit there and say, but wait a minute, aren't those childhood diseases and don't we have vaccinations, et cetera? Well, we did talk about this before. The herd immunity. And in some places in this country, and not only ours, but for example, I remember hearing cases in Great Britain where the herd immunity, the number that have to be vaccinated, well, they've fallen below that. And therefore, the population has a much greater risk of having an outbreak. So if you look at the process stage, first you have, of course, your data collection and pre-processing. The output is that you're going to get a real-time data for early outbreak detection. Then you start bringing in the computers, which are fantastic in the sense of number crunching and data analysis, and you can bring in spatial analysis or using geographic information studies, and you'll see this signal and something unusual noted. When you have the epidemiological analysis and interpretation, you can come up with some very interesting information about the outbreak, such as if you have this noted here, high probability outbreak, there's one way, a low probability of the outbreak, or that you have an error. There was a reporting abnormality and data processing error. This happens sometimes. That's why sometimes numbers are checked, data is collected, some of it has to be recollected. Okay? Finally, as you do the investigation, and the investigation may be epidemiologic or even forensic, you're able to winnow out, almost you want to say, the needle that is in the haystack, and you get the true increase in disease. You may be able to determine if instead it's really a natural variation in the disease, there's no real threatening outbreak, or you have a natural outbreak or a deliberate outbreak. Now, for those of you that are wondering why are we bringing in the forensic, there has been at times some suspicion that, for example, Covars was a developed biological agent, or just that you can sit there and say certain people were sick, refused to get further treatment, and went out into the general population. And so now you have to start tracking down where they've been, etc. Well, let's continue here. Now, the steps in an outbreak investigation. You confirm the existence of the outbreak. You verify the diagnosis, define and identify cases, manage and analyze the data, develop, test, and reevaluate the hypothesis, implement control and prevention measures, and then communicate your findings. Now, if you're a real big sleuth or a real got in your blood sort of like, I want to hunt things down and do that, epidemiologists are really good at this. They really like hunting things down and connecting the dots. So it's not just confirming, yeah, we've got something here, but you have to verify it. How do you verify it? Could be by various tests. It could be by various signs and symptoms as well. You may have to define and identify which cases they are. Some people may say, gee, I don't feel very well, but it's really a matter of, okay, they got food poisoning. Okay, they've got a cold or the flu. And that was partly some of the situations that occurred when we were talking about COVID. Some people would wonder, my gosh, I'm not feeling well, etc., and take a COVID test. And, well, COVID tests came back negative, and what they had really was the flu. Now, after you've analyzed the data, you start to develop, test, and if necessary, reevaluate your hypothesis. And if you think about it, when you were reading the information on John Snow, who was the father of epidemiology, he had to come up with collecting all of his data. He had to analyze it. Now, this is in the days of paper and pen, okay, or paper and pencil. I wouldn't say pencil even. Mostly used pen. But you also have to keep in mind several things. Snow's work was still with the possibility that one of the theories was that disease is caused by bad air, the miasma theory. The germ theory had not been established and completely accepted yet. That's one. Two, you had a lot of people dying, and in some cases they would die very fast. This is in COVID. Excuse me, not COVID, it's cholera in London. And sometimes you have to wonder, gosh, did I miss something? That these people go to different places besides that well. Then you're going to implement the control and the prevention measures. Well, the control was basically removing that handle. So that well, people weren't drinking the well water, which turned out to be that it was the well, the spring underneath was contaminated by sewage. Okay? And then you want to communicate your findings. Now, it's important to communicate it because several things. One, the public can understand what are some of the control measures. And they can also understand why certain things are going on. Two, you communicate your findings because there are other doctors, not only, let's say, in the country, but in other parts of the world, that really have the same situation occurring in some cases. And three, communicating the findings will usually lead you to another question that you can then investigate and do research for it. Now, that seems to be pretty obvious, but for some people it's not. Now, during the steps of an outbreak investigation, you have to start asking yourself, well, what's the etiological agent responsible for the outbreak or epidemic? What is or are the predominant modes of transmission? So is it air, water, food? Is it by sneezing, etc.? There was a researcher who found that cholera could be transmitted not merely by contaminated water, but by contaminated food. Now, what specific sources of the disease can be identified? Sometimes it's relatively easy, such as with John Snow finding that source was the well water. What specific practices or environmental deficiencies have contributed to the outbreak? Okay, the practice, we'll take that as an example first. The prion-based disease Kuru, which was human-to-human passage, the practice was basically by this New Guinean population of natives. When someone died, they basically were going to have the funeral, and part of the funeral was to take and handle the deceased brain and pass it around, and in some cases take a piece of it. Unfortunately, what you're doing is infecting yourself with the same neurodegenerative disease that caused that first person to die. Now, environmental deficiencies. We have a lot of them. I just gave you the one about what John Snow had to deal with. There are some really good books about it out there, but here's the thing that you got to keep in mind. That's why we're so picky today with where water comes from, whether it comes from municipal areas, whether it comes from wells. But if it comes from wells, you really have to test to make sure that that well doesn't get contaminated, particularly with even fluid from runoff from septic systems, because that's where you can have this cycle going on. Also, what is the chain of events that led to the outbreak? Now, there are a lot of possibilities of when you talk about a chain of events. That's why they have to go out and use, I would call it the shoe leather approach, walking around asking who were the contacts, etc., till they can probably come up with the patient zero, the person that first got the disease. How did they get it? And how did they spread it? In the early part of COVID, one of the disasters that happened, and they really didn't know about how serious this was because the Chinese government would limit the information. So there was a company in Boston called Biogen. They had a big conference of some, and they brought in like 1,300 researchers, etc., from all over the place, all over the United States and different parts of the world. Several of them came in with COVID and to spread it to others. And those people went home all over the U.S., all over Europe, etc., and you can see where the picture is, and they spread it. Okay? Now, another question is, how rapid is the progress towards specified goals in reducing disease incidence and transmission? If you implement certain strategies, how fast will you see a decline in that disease? If it's a matter of yellow fever, and you're able to completely dry up all the areas where the mosquitoes are, which might transmit yellow fever, you might see the progress turn around relatively quickly. In other situations, it might take some days, it might take weeks or longer. Finally, what are the future public health priorities, policies, and strategies that are based on the data? Now, remember, the data is collected, and it will tell you, okay, how serious is disease? What do we need to do if we have it? What are the strategies that we need to reduce the chance of it being spread? If you remember, the public health people, when COVID first started rising up, you had meetings where people were being told, you got to wash your hands so much, and you've got to, if you feel like you're sick, you stay home, and it went on and on and on. And that, in some cases, worked very well for reducing the incidence. Now, when we talk about public health surveillance, this is really seeking to meet a variety of objectives. These will include the detection of the outbreaks, but also documenting of the distribution and spread of the disease, monitoring for changes, detecting changes in public health or healthcare practice, and generating hypotheses about the cause of the disease. Now, some people, if you've forgotten the entire concept of the scientific method, hypotheses are never proven. They're supported by the data. They can be disproved. And so it turns out that certain individuals will get sick, and then it's first thought that's because they had, bad strawberries. And it turns out it was bad raspberries. I'll give you an example of that. There was a disorder, cyclosporia, that ended up really showing up in this party of 200 at a wedding. This was some years back in the 90s. It was very sad. And they had this massive fruit bowl, and it had all sorts of things in it. And of course, it was set up in such a way that the watermelon that they had sliced open was part of almost like the bowl. Well, investigators got in there and they asked all sorts of questions. Did you do this? Did you eat that? Did you do this? Did you eat that? And they tested some, I think they had some of the samples left. And it wasn't the strawberries that they first thought. It turned out to be the raspberries. And when they went back to the supplier of the raspberries, they said, well, we got it from, it was imported. Well, where was it imported from? Turns out it was Guatemala, Central America. And basically out there in agricultural fields, they don't have porta potties. They don't wash the hands. And the people that come in sort of work a while and leave. The best hypothesis to this day is that it was an individual who was traveling around the world. The individual was travel, run out of money, do work, like in fields and things like that, generate enough cash and leave. But in doing so, they basically spread the disease. Now, to help you to understand something, this particular organism is normally seen in India and Nepal. So for it to show up in Guatemala, everybody was scratching their heads going, what the heck's going on here? And they think that that is what happened. Do they have the name of the person or anything else like that? Nope. This person probably continued on their journey around the world, had fun, basically would work till they ran out of money, would basically travel till they ran out of money and then work in the local fields and farms, etc. But they had a parasitic disease in them. And they didn't clean themselves off properly, and so they spread that to the food that they were picking that was eventually shipped up from Guatemala to the United States, from the United States to that particular wedding planning, wedding catering service. So the connections that we have today as a global economy, etc., means that outbreaks can pop up. And the questions are, well, why is it popping up here? That's not normal. Okay. Now, to collect data, various means of collection and monitoring may be necessary. Here's one thing to keep in mind. There's what is known as syndromic surveillance, and that may use a constellation of symptoms or syndrome instead of a disease as an early warning system for whether there is a sudden or unusual increase in the disease. So when we talk about syndrome, they might have, spiking fever and petechiae, which look like little bruises all over the skin, and not come to the conclusion that it was Rocky Mountain spotted fever or something else like that yet. Now, sentinel surveillance is a little bit different. This enrolls a small number of health facilities reporting on health events generalizable to the whole population. Events of interest are collected only from this sample of sentinel sites, which may provide an early warning signal or a red alert for a particular disease. So what you've got really is the syndromic takes a big picture, but it depends, A, on whether the parties involved are going to participate, and B, it's only looking at symptoms. The sentinel surveillance takes a small number of health facilities, and they're sort of considered sort of like a reflection of the whole population for certain diseases. Now, you might be wondering, well, why don't we have everything reported? It's an interesting thing I found out years ago when I was working in another facility. We do not have socialized medicine, and as a result, every number of broken fingers and broken arms and runny noses is not recorded. There's a set of about 10, maybe 12 hospitals in the nation that they use as sort of the sentinel surveillance system. They sort of dip their toes in, give us what we need, and that's it. Whereas if you go to some other socialized medicine countries, Canada, UK, etc., they'll report everything. We've got 15 people that had crushed toes. We've got 467 people who lost their eyesight due to diabetes, and on and on and on. The problem is, it's a different way of how we look at things. Congress mandates that about 20 communicable diseases are to be monitored by the CDC. 20 communicable diseases. That's it. So let's continue to move on. Now, when we collect data for the diseases, these two factors are critical. First, sensitivity, and the other is specificity. Well, what's the difference? Sensitivity is the proportion of cases correctly detected or tested positive. A higher sensitivity implies fewer false negative results. So pregnancy test kits, COVID kits, all these other type of detection kits out there, they really have to have a fairly high sensitivity. And this is partly done through the FDA. The FDA. They have set a criteria so that you have to have a very high sensitivity. Now, a specificity. This is the proportion of healthy individuals who are correctly detected negative or tested negative. A higher specificity implies fewer false positives. The way to look at it this way is this. Sensitivity is able to detect effectively all the correct positive cases and have very few false negative results. Specificity is able to accurately determine which patients are negative, and thereby you will have fewer false positives. If you're talking about like a pregnancy test kit, nobody wants to have a false negative result, and nobody wants to have a false positive result. Now, positive predictive value. This is the proportion of cases who have truly positive test results or a test event out of all those who've had positive test results are detected. Now, negative predictive value, NPV, and this is a proportion of healthy individuals truly diagnosed as negative out of all those individuals who have a negative test result. So positive predictive value, absolutely confirmed. You can give it several different tests. You can test it in other ways. They are positive. The same thing, but opposite for the negative predictive value. Now, with persistent infectious diseases, public health threats mandate clear and standardized outbreak response and surveillance systems. I'll give you an example. It was, I'm going to say it's about 10, no, or more like 20 years ago. Just before school was going to start, and it was a hot week in Western Mass. And in Western Mass, unfortunately, they started having an outbreak of Shigella with little kids. Now, the problem with that is, with little kids having this, that means that eventually adults are getting there. And so, of course, you have the diarrhea and everything that goes with it. That means that you have to do cleaning of hands, etc., because anytime that you touch an inanimate object, a fomite, you could be leaving behind some Shigella bacteria. And also, if you notice, I said it was coming toward the end of summer. That means that the school systems were facing the possibility of having kids come in, and within a short period of time, they were afraid that kindergarten or K through 8 schools would start having serious outbreaks of that. And so a lot of cleaning was necessary in preparation for that. Okay? Because this was a very clear, potentially threatening situation. And public health would give its input on how to basically make sure that the whole area is cleared up, etc. As it was so, there was usually when you start getting into September, you start having all these festivals and events. Some of them are agricultural based, some of them are large events that cover most of the New England states, like the Big E. There was one that was called the Polish Festival, because of the area spring of Chicopee that had a massive amount of its population of Polish ancestry. Now, it was the first time I've ever seen it where you go to the porta potty, you come out, and they had brought in from the nearby military base these sort of portable sink with soap, and they had even a water container truck or trailer. And they just wanted to make sure that they weren't having this being spread out in that festival. So, epidemiological expertise is needed from the design of data systems to using a variety of types of health information and data sources to analyzing, interpreting, and communicating complex data. So when one sits there and thinks about an epidemiologist, the days of just one individual are sort of out the door. There might be one individual that does the contact tracing or the investigations, but hands back the data, and the data goes through basically electronic data analysis, electronic information dissemination, analyzing and making sure that the diagnosis that there is infection here is absolutely correct. Let's take a look at this for a second. We have the electronic case reporting, ECR. And how does it work? And this is within the CDC, by the way. Patient is diagnosed with a reportable condition such as COVID-19. The healthcare provider enters the patient's information into an electronic health record. Okay? And so from there, the data in the EHR automatically triggers a case report that is validated, sent to the appropriate public health agency if it meets reportability criteria. The public health agency receives the case report in real time, and a response about reportability is sent back to the provider. Then, the state or local health department reaches out to the patient for contact tracing services or other public health action. Now, if you think about this could happen at 1, this occurs at 2, and then these are occurring in the matter of less than a couple of hours. It's almost as if, okay, fine, the patient goes home and then gets a call from the state and local health department. And this is really fantastic compared to what we used to have for problems and for epidemiological detection. Now, we have here again the disease burden pyramid. We have that disease pyramid we talked about last time, but what you have are basically for many of the diseases, this is particularly if they're foodborne diseases, where the symptoms are going to vary from mild to life-threatening and even death. The cases that are reported are only a very small percentage of the actual number of cases that occur. And I'm sure you probably know of situations. Let me just take food poisoning, for example. And please excuse my dog in the back there. Where somebody goes and they go to one of the fast food joints and they have something and they head home and they feel terrible. They've got diarrhea or vomiting or whatever. And within the next day, they're up and fine. Do they report it? Probably not. And this is part of the problem today with certain illnesses. A healthy individual might shake off within about a 12 to 24 hour period that ailment. But what if they're elderly with a suppressed immunity or immunosuppressed, either because they've got an immunodeficiency disease or they are under some treatment for some ailment that suppresses their immunity? Well, if you look at this, you've got different surveys. Exposed individuals and ill individuals are part of the population survey. The provider surveys are the care-seeking individuals. They are also the ones getting the specimen and testing it out. That's where you see the laboratory survey, which has completed the analysis. And the lab confirms the cases and then sends it out. And then you have reported cases. And that's where you have to bring in the surveillance. Or that's where the surveillance may be occurring. The problem we have is certain diseases don't always get noticed. Sometimes because the individuals are afraid. Sometimes they are just not giving it much credence. And sometimes they don't understand what it is until much later. There we go. Now, the public health surveillance system for disease reporting. You have to keep in mind that the case surveillance flow is going to go from local to international. And what do I mean by that? I'll give you an example. It was 1995. There was a place, a little restaurant, Two Brothers. Now, they had, of course, other help besides those two brothers. Anyways, you had some very interesting situations occurring where they got very popular. And so and this was January, and so you had hockey teams, you had the pee-wee soccer teams and all this. Everybody would come over, for pizza or grinders or a salad or something like that, And then something happened. And they had to go through and investigate. The epidemiologists, they found out, well, one of the individuals at that facility got hepatitis A. And that meant that a whole mess of people, ready? 1,000 people in the town had to stop by and get injections of basically IgG. Now, this is sort of prophylactic, so you don't get hepatitis A. Now, for those of you that are not aware of this stuff comes out, it's very proteinaceous, so it comes out like oil, which basically means, no, you are not going to get the injection in the arm, you are going to get it in the buttocks. And I'm got my head to the wall, and I'm the nurse is doing it, but, they have these privacy curtains. And next to it, I can hear this poor little 3-year-old screaming and his brains out going, Now, all these people, 1,000 people had to get a preventive to stop what could have been a very serious outbreak. And so I had a few minutes, and I was chatting with the nurse there, and she said, "You want to know the biggest thing? We found the guy at that restaurant that was making stuff." Now, let me premise it also with this. Nobody wore gloves, and if they wore gloves, it was to handle the cash. So they were protected from germs from the cash. Big deal. But when it came to making the pizzas and making the grinders and everything, they had no gloves. So the nurse, public health nurse, told me, she said, to this day, we still don't know. We found the number one guy. He went to a party and he said he'd gotten it there. But nobody knows what he was doing there to get that infection. So we don't know if there were other outbreaks somewhere else because that infected person passed it on to this gentleman who eventually worked at the pizzeria, and so forth and so on. Now, any conditions at the local, state, etc., they're passed on. The cases are reported to hospitals, healthcare providers, laboratories, etc. This is all based on the state reportable conditions. That's the state side, okay? But then you're going to have that information passed over, if necessary, to the Centers for Disease Control and Prevention. That's sort of the central nexus for the United States. And if they need to, they will send in epidemiologists, etc., if the outbreak, if the disease is very, very serious. At the same time, CDC has a public health event that may be of international concern. They will pass this information to the World Health Organization. And so this is the national, international notifiable conditions. Just to give you an idea, they had, I think it was about 2008 or 2009, an outbreak of bird flu. And it kind of burned itself out, fortunately. But the situation was such that I remember listening to the head of WHO, and he said, basically, if it occurs in a country, it's an epidemic. If it goes passes the borders to other nearby countries, then we have a pandemic. And that's the simple definition you might want to keep in mind. Once you breach basically the containment of the national border, you don't know where it's going to go. Okay, let's move on. Now, if we look at intervention points across the chain of infection, I like this chart because it really gives you some thought, okay? Intervention measures. Let me give you examples of this. For the reservoir, now this is in the environment side only. We've got the host side also. You want to disrupt the transmission route, you disrupt the reservoir. An example, in certain places, they would have an outbreak of rabies in all of the raccoons and stuff. So what would be done in cooperation with the Wildlife Association, the wildlife agencies of that state, is they would fly over areas with helicopters and drop pellets of food that would draw and attract those raccoons. What was in those pellets was a rabies vaccine. So when they consume that, they get themselves vaccinated against rabies. Okay? If you can look at the intervention methods against the source might be a carrier, or it might be contaminated water. So that's where you have intervention measures such as announcements: boil all your water. We can't guarantee it's going to be that. Or transmission routes. So we shut down all the water transportation within that area because it is contaminated, and we'll let you know after we flush the area with chlorine or whatever. Now, when we come into the host, remember that the host has both the innate and the adaptive immunity. The innate immunity includes the barriers such as skin, such as acidity in the stomach. It will probably make certain proteolytic enzymes either in the skin, in tears, etc. But if you have an infection that's going to breach that, you have other options, and such options might include interventions like I was mentioning, a prophylactic vaccination, etc. Now, for specific immunity, what we're talking about there may be more adaptive immunity. And in that case, that's where you go. And for example, some of the intervention methods might be a large number of students have been exposed to bacterial meningitis. So you just line up everybody and give them a prophylactic antibiotic before there's a chance of it spreading. We have the possibility of hepatitis B. Then you have people line up for hepatitis B vaccinations. Now, in the chance that you do have an infection and you have the specific immunity being affected, then you have to work with recovery. Sometimes the recovery will be other drugs that can stop viral replication, bacterial replication, etc., and give the body the chance to fight off that infection and move into the recovery stage. And finally, unfortunately, we come to the possibility of death. A lot of disorders that we're going to walk into, death does occur if there is no treatment. Carriers sometimes do not face death, but rather continue to keep spreading the disease. But intervention measures against a carrier would be very important, and that sometimes you find out who's the patient zero, and you have to make sure that the person's the patient zero. So you do a lot of who did you see, where'd you go, what'd you do, what'd you eat, what restaurant were you at, etc. You try to track down all the steps of the carrier and perhaps even determine how they were able to originally pick up the disease. Okay? Now, let's talk about for a few minutes the methods of detection. When we talk about the methods of detection, usually we've got to get an analysis of a patient's sample. And those samples include blood, sputum, urine, cerebral spinal fluid, and stool. Each one of them may contain various pathogenic components. For example, stool, if we're talking about hookworm or tapeworm or something else like that, may have their eggs in them. Blood may have malaria present. It may have viruses present. It may have bacteria present, etc. Sputum may contain bacteria that can withstand the sputum, and that might include things like tuberculosis. Okay? Whereas urine, sometimes everybody focuses on the urine chemistry. So what's the pH? What's the amount of sodium, potassium, the electrolytes? But you do from time to time get some evidence of an infection. You may see leukocytes, you may see nitrites, and sometimes you will see some bacteria that are causing a urinary tract infection and things like that. Now, for cerebral spinal fluid, this is a little bit different. The cerebral spinal fluid, sometimes you will see signs of a virus. You have to understand something. CSF looks like water. It's clear, transparent. But if there is some infection, you may start seeing other substances present in the CSF. And you can detect them with certain tests. So here we have the sputum guide. It includes the 1, 2, 3, 4. How do we collect it? We have over here someone who is going through providing a blood sample. We have over here 3 types of urinalysis. Now, urinalysis with the microscope, they're going to be looking for any of the following. They're going to be looking for crystals that are in the urine. They might be looking for what they call casts, which are basically components of the cell lining. Or in some cases, they may actually see bacteria, so they can do a stain. There also are, of course, rapid urinalysis tests that will tell you a variety of conditions, the pH, the sodium, the potassium, the BUN, etc. And then again, you've got the 24-hour urine collection, where you basically have provided the urine, and it's going to be done 24 hours, and then sometimes we'll give a clear picture of the of the renal function. So is the kidneys working properly? Here you can see at this far end here, down in the lumbar area, a cerebral spinal fluid tap, or a spinal tap, as they would call it. And basically what happens is the spinal cord stops at a certain point and just becomes loose nerves. But you still got a large area that is somewhat filled with cerebral spinal fluid, makes it easy to extract. You have very little chance of damaging nerves. And you can use it also for doing epidermals, for surgery and things like that. And finally, as you can see here, this would be some of the items you would see in a stool sample. Stool sample, they would be looking for a variety of the different types of eggs. Rarely do you get to see as many living organisms, although you can sometimes find that, whether we're talking bacteria or amoebas, etc. But when it comes to something like helminths, in other words, one type of worm or another, 9 times out of 10, you're going to see that in the stool. And that's where you're going to be able to make a diagnosis. Now, what about laboratory methods of detection? Well, I showed you a few of them right off the bat, the microscope, the staining, etc. But we can go beyond that. Today, we have all these tools that allow us to rapidly analyze situations. For microbial analysis of the sample, you can do polymerase chain reaction. Now, by the way, PCR can be done to detect fungi, bacteria, viruses, and it's basically looking for certain markers in there. We also have ELISA, which stands for the enzyme-linked immunoassay. An ELISA test, there are different versions of them, but these are the ones that are relatively quick. For example, you may have seen an ELISA test if you came in and were suspected of strep A. They just swab in the back of your throat, put it into a small little plastic structure, and if it reacts, then you've got that particular strain of bacteria. Now, whole genome sequencing, WGS, that's not done every day for everybody, but it is very useful for the detection of pathogen, genetic variants, gene defects, and foodborne diseases. So when we get into some of the foodborne diseases, we may mention some of that. One of the things that happens is this: you get a sort of strange disorder, and you would think it's a type of disease, etc., but it's not responding to certain treatments. And doing a WGS, you come to find out, lo and behold, it has extra DNA inside, let's say bacteria, that provide it with microbial resistance. Microbial resistance we're going to cover later on, but this is really critical. We are seeing diseases that are coming to the point of almost no treatment at all available. There's certain strains of gonorrhea right now, completely immune to any of the antibiotics we had. We're seeing fungal organisms, etc. And when you do a whole gene sequence, you can identify those particular sequence areas in the genome that will tell you, yes, this is a strain that's resistant. And so you're going to have to find other ways to treat it. Then we have, of course, the microbial cultures, whether you're culturing fungal cells, bacteria cells, and also it's not a matter of just, well, let's put in gram stain, or what's the size and shape of the cell. But you're going to be able to do a lot of other biochemical tests. And these biochemical tests, taken together with information such as, let's say, bacteria, the cell shape, and its staining capability, you will be able to sort of put this together almost like in a dichotomous key or almost like in a formula, and boom, you have an idea of what type of organism it is, where it's been normally found, and what is it resistant to and not resistant to. Now, here you have a situation where you have the PCR. So a sample is taken, and in this case, I think this is viral. So what we do is we take the viruses and we basically deactivate the viruses and we extract out their RNA. Then we add a particular protein called viral RNA retrotranscription. That will convert the RNA to DNA. From DNA, we'll then go through an analysis where basically you do these cycles. And basically it's a cycle that identifies that particular sequence, makes new copy, and in a short period of time, we're talking maybe 30 seconds or so. Then we're talking about another 25, 30 seconds where we separate them, okay? And then we allow them also to cool down and anneal, come together again, and we continue this cycle for so many times. Think about it this way, as I used to describe to my biotech students. You've heard of someone saying, well, it's like finding a needle in a haystack. But what if I included more needles in my haystack? Then I'd have a higher chance of finding them, wouldn't I? Okay. And the needles would be that particular sequence that you're looking for to identify that particular viral disease. Now, in this one, this is whole genome sequencing. And what you basically do is you take the whole genome, you're basically going to make many copies of it. And then you are going to break it down, analyze all the A's and T's and C's and G's, etc. As a result, you will also be connected to a large database, and you can say, well, this is a fungi. This is fungi A114. It looks like a penicillium type, but it does not give penicillin. Rather, it will cause fevers, etc. If you take a look over at this image, these are fungal cells. So this is a microscopic method of identification of these type of cells. And they've added a particular stain that's unique for fungal cells, which is kind of blue. Here we have bacteria. Now, if you notice, you're going to have two different types of bacteria here. You can tell that because of the not only color, but the shape. You have the cocci type, they're roundish, and those are going to be purple, so therefore they will be gram-positive. The long rod-shaped ones, these are gram-negative, and so you have basically two bacteria here. One stains gram-negative, so it's pink, it's rod-shaped. The other one stains purple and is oxy-shaped, in other words, round. But you know what? That doesn't do enough. We needed to know more. And if you see down here, this is where this gentleman is giving a discussion on growing the bacteria on different types of medium, and they will have certain biochemical characteristics as a result. Some of them will react in a particular way, and give off certain enzymes into the surrounding medium. There are other situations where you will have to just add a few drops into a culture tube and wait 24 hours, and then you will have a certain reaction. Does it ferment? Does it not ferment? But all of this information brought together will give you a laboratory confirmation of what type of organism this is. Now, how else do we control disease? Wow. You know what? There's some of the things we take for granted. We have antibiotics. They go after bacteria. Antifungals, and they go after fungal organisms. Antihelmetics. Now, antihelmetics are only for those organisms that are of one type or another, type of roundworm, flatworm, fluke, all of these that can cause disease. Let's say water's been contaminated and you want to access it. One way to prevent waterborne diseases is disinfection of the drinking water. You might use chlorine, but as one city found out, chlorine didn't always do it, so they had to add on another substance. In this case was ozone. Don't worry, ozone's not going to hurt you, and basically what happens is it passes out, but it will kill bacterial organisms and some protozoans. Other cases, you use ultraviolet light. Particularly, ultraviolet light will break down DNA and RNA. Works out well. You could be cleaning up your work or your eating areas, and that is necessary to be part of the control of disease, whether you're talking about hospitals, or you're talking about the air that people are breathing, or fomites, or clothing. And when I say air, there are certain places where basically you've probably all heard of clean labs when they make these microchips. Well, it's because they don't want dust onto the chips as they're working on them. Well, the same thing. You have places where individuals are working, or people that are doing surgery, for example, and the air has to be really clean. And so it gets filtered and gets removed particles down to a certain size. Fomites, if you remember, these are non-living organisms, non-living structures that basically may be a surface or a doorknob or a handle that somebody may have put their hand onto, and unfortunately, they've left behind bacteria that makes people sick. Removal of outside trash. We're going to actually do some discussion about that in so many weeks. Rats. Why am I saying rats? Well, if you go to New York City and you go to the subway, it's not unusual to find them. If they have a trash removal strike, it's worse, because what the people basically do is they take their trash bags and put them outside onto the sidewalk, and suddenly they pile up, and of course the rats sit there and go, let me go in there. I can get pizza, and I can get old this and old that, doesn't matter. Mosquitoes can transmit yellow fever, malaria, and a whole host of other diseases. Mice, similar to rats, they can pass on various organisms, even from their urine. Leptospirosis is one case. Fleas. Well, fleas are the best known vectors to pass on such horrible diseases as plague and a variety of other diseases through their bites. Okay? And we'll discuss that later on down the road. And just another method of controlling disease. How many times are we taught, wash your hands? Wash your hands, wash your hands, wash your hands. And not just get them wet, use a good disinfectant soap. And you can see this here. Oh, by the way, there are drugs that are antifungal. Here's a couple of the molecules. This map shows you a basic bacteria and where the different types of antibiotics target, whether it's the DNA loop itself, ribosomes, the movement of the flagellum, punching holes into the cell wall or cell membrane or both. Then we come to water treatment. Now, lots of people will say, well, we've got a river nearby. Yeah. But you don't know what's been discharged into the river water upstream. And so what happens is, to guarantee that no one gets sick, the river water gets treated. Any floating material, even small particles or organisms, gets captured by coagulation. Then we have sedimentation, and we send the sediment out. We have filtration here. Then we have disinfection. Now, the disinfection area, that's where they may use chlorine. They may also use chlorine and UV light or ozone or both. And then we have, of course, storage and then use, which I think is pretty good. Here we have air filtration systems. Now, some of you may have heard of a HEPA filter, high efficiency particle air filter. And basically, these are filters that basically will get very, very, very small particles. Not just bacteria, pollen, stuff like that. But some of them will go much smaller than that. Smoke particles and some viruses, or at least the dust where microbes are rolling around on. Here is one of the nastiest beasts on the planet, and that is our mosquitoes. And as you can see here, we have been trying to regulate it. Now, just to bring up one point to help you, the mosquitoes today, depending on where you are a little bit more challenging. The tiger mosquito does not just feed at night, it feeds during the day. So it's conceivable that one can be in some of the southern states where it's present. It came from I think it came from either Southeast Asia or Latin America. And unfortunately, it will carry all sorts of different diseases, encephalitis and malaria and yellow fever and dengue fever, etc. And the problem is, everybody would think, well, it's daytime, it's not going to be a problem. Yes, it will. And they have to sometimes go looking for all the trash, half-filled cans or containers that have picked up rainwater that's now a breeding zone for those mosquitoes, etc. Here we have two happy little mice chewing on a chicken nugget. Yep, and you notice it's daytime. They're not hiding. They don't have to. And then we come up into the different means in which individuals will have transfer from the infected individual to the susceptible individual. So we're talking about airborne viruses, inhalation of the spray when someone coughs or sneezes. You have airborne, such as when you're talking with someone. One of the interesting things is that sometimes the particle that gets expelled may begin to dry, but the organisms that are on it may still be infectious. We have surface viruses, and we have all sorts of other means of suspension and being picked up, nose, mouth, by touching, inhalation, etc. So, from here, I wanted to bring up the issues about vaccines. Now, there are various types. Some of them have limits of efficacy for their lifespan. And what I mean by that is, I'll give you an example. Hepatitis B, when they first started distributing in about 1985, they basically said you needed a booster every 5 years. After the first 5 years, I got my booster. Second time around, they just tested me for whether I still had the antibodies, and they said, yeah, you have the antibodies. You don't need another booster. There are some vaccines, such as the pneumococcal one, that one and done. That's it. But also vaccines, you have limits of the patient's age and immune status. If they're very, very, very old, they may need it, but it may take longer for that patient to develop the immune response. We can't give infant newborns a vaccine. And you say, why? And you say, simple. It takes about 6 months for them to develop their own activity of their own immune system. So it's usually 6 to 8 months of age before they start getting some vaccinations. Now, also one other thing. If someone has some state of immunosuppression because of disease or treatment, the problem with some of the vaccines is that the vaccine may be an attenuated, in other words, weakened virus, weakened bacteria. In a normal person, they would develop the immunoreactivity. But in these individuals, the attenuated organism may actually convert back over to a wild type. In other words, causing them the illness. Okay? Now, in vaccines, you add adjuvants. Now, an adjuvant is a substance that increases or modulates the immune response to a vaccine. Now, adjuvants can be divided into delivery systems. Are they particle or immune potentiators, such as immunostimulatory? Some of the examples of adjuvants, and some adjuvants were actually also used in part as preservatives. But usually there are preservatives that are separate. But most adjuvants, you'll see some type of, and it's aluminum salts, not sales. I apologize for that. It might be an oil and water emulsion. There's one that has picked up some attention, QS21 saponin. Now, a saponin is a type of, you almost want to say it's like an amphipathic molecule. It's like a soap molecule. Think about it. And some of these have been extracted from trees, and they have found to be very useful. And since the population is generally trying to get away from aluminum injections, this seems to be something that's going to have a future, this QS21. Now, how is this monitored? The entire issue about vaccines is not that, and believe me, I've heard all sorts of stuff like, oh, they're going to have some alien implant and all that other nonsense. Okay, look, there is a group called ACIP, the Advisory Committee on Immunization Practices. Now, this is an advisory committee. but it's part of the CDC, Center for Disease Control. And what ACIP develops is recommendations for U.S. immunization. So these are the people that help sit down and say, okay, fine, you're going to get your measles, mumps, and rubella at age 1, 5, 10, etc. Just as a hypothetical situation, okay? They include the ages when the vaccine should be given, the number of doses, the time between the doses, and precautions and contraindications, as well as limits to the patient's age and immune status. So what do I mean by this? A very simple situation. When I first got hepatitis B vaccination, the first shot was, I'll call it day 0. 30 days later, I got my second shot. And then 90 days later, from my initial shot, I got my third shot. Seroconverted very nicely. A lot of people seroconvert, meaning that they now have all the antibodies, etc., by the second shot. But to cover everyone, that's when they bring up the use of a third vaccine. And if you notice there's a signature of one of the authors of the books we use here. And he's giving a shot to the little boy. Where did vaccines all come from? Well, here we go. The father of immunology was Dr. Edward Jenner. He created the first successful vaccine. He based his observations on that the milkmaids or maidens, the women that basically go out and milk cows, may get exposed to cowpox. It's different from smallpox. But then he noticed that their skin was very nice, very clean, no pock marks, etc. So he said, maybe there's a relationship between smallpox and cowpox. Okay? And that's why they don't get smallpox, because they've already been exposed to the cowpox. So in May of 1796, this English physician, Dr. Jenner, expanded on this discovery and inoculated an 8-year-old boy, James Phipps. And basically he had taken the matter collected from a cowpox sore. And you can see the maid right here wrapping herself with a little gauze or whatever. He took it from the hand of the milkmaid, and then he injected it into the boy. And he was successful. The boy did not get smallpox. You have to understand, on average, until smallpox was eradicated, it would cause morbidity in 40% of the people who were sick with it. Mortality in 40% of the people. Morbidity, everybody got sick that was exposed to it, unless they had been previously exposed to it. But it was still a very nasty disease to have. Now, here's a couple pointers that I thought I'd bring to your attention. Here are some of the common vaccine components: stabilizers, preservatives, trace compounds, antibiotics. Sometimes they've had to do that. Adjuvants, and then, of course, the biggie, your active ingredients. Vaccines can be attenuated bacteria or virus. They can be destroyed, killed virus or bacteria. Okay. They can be just the proteins on the outside, protein subunits. They can be the viral-like particles that basically you take the protein subunits, you attach them to sort of like a liposome. A liposome is sort of contains a membrane, and that's about it. Now, the biggie that caused a lot of people to get upset was doing some of the DNA or some of the RNA and injecting it into a human's muscle. The genetic information would pass out protein subunits, and that's how the individuals would be resistant. There were some people that insisted that they were going to have all sorts of other things happen, and they would be genetically modified and all this other stuff. Now, you have to understand something, that the FDA does not approve stuff, and they have open meetings where a whole group of the doctors review all of the data, which I'm going to show you in a minute how this is standard goes. But moving aside of SARS for a second, we go over to the vaccine rates of children entering kindergarten in 2020 and 2021. Okay? Take a look. Here we have diphtheria, tetanus, pertussis. This is in Louisiana. Compare this to the United States. Hepatitis B, we compare that to the U.S. Mumps, measles, and rubella, that's the MMR part. They combine it, and you have that 76.2 versus 93.9. And then polio, 98.6 versus 93.9. It is extremely important that we do this because these children otherwise would have some of these diseases, disorders, and in some cases, death. Also, some of the diseases, and we'll go into it later on in the semester, can have an adverse effect on other individuals, such as pregnant mothers. What happens is with one disease, and I believe it's the measles virus. No, excuse me. It's the German measles. What happens is, let's say it's a young lady and she's working as a teacher, and the kids come up to her, and of course she's pregnant. If she gets the virus will alter the development of the developing embryo. In this case, usually developing fetus, and the children will end up with massive defects in the proper structure of the heart, the lungs, the eye. They may be blind, et cetera, because these viruses will zero in on certain neural tissue and several other tissues. And so you end up with either stillbirths or miscarriages, or baby is born and then dies very shortly afterwards. So there is direct consequences to populations that choose not to vaccinate. So let's take a look at some of the impact of vaccinations. So we have an RO here of 6. It's a hypothetical disease. 90% of the population has been vaccinated. Now, every one of the oranges you can see is susceptible. There are also tertiary cases. But if you notice, all these susceptible ones have been given a vaccination. So, what they mean by the index case, that's case patient 0, sort of. Secondary case, there's 1, and that's it. So in other words, you've taken a disease that went RO6, and you've converted the condition, so now it's almost like an RO1. But without the vaccination, what would happen? You have an RO6, right? Here's our index case. Here are the secondary cases. And then you see all these light orange. These are all the secondary, the tertiary cases. But wait, we still have more. We have more susceptible cases here. Notice the green. Those are the quaternary cases. So in other words, think of this as 1, 2, 3, and then 4. Quite a lot of people made sick. So if you see this, you have the left panel, and it shows the impact of the vaccination. If 90% of vaccination were achieved, you want to note also that the people in the upper left-hand corner do not become infected despite not being vaccinated. Okay, up here. Because what do they have? Basically, everyone here has been vaccinated. Okay. Now, how do we go through the phases of vaccine development? It is very similar to the process for drug approval. Okay. And I used to advise a lot of companies about this, so we're going to go through this. Prior to the submission of an investigation new drug, this is the documents that say, hey, this is what we have. You have to, of course, go through all sorts of exploratory and preclinical stages. So you might include animal testing. You might have some basic science research into the potential natural and synthetic antigens. What you may also do is see if in a tissue culture this might cause some positive effects, etc. All of this data is brought before the FDA, and you file what is called an investigational new drug. Now, investigational new drug is not something that, okay, I handed them, what are you going to do now? You've got to have your plan already. So you have a candidate vaccine that shows promise of safety and efficacy. So now you're going to do clinical trials. Now, first off with a clinical trial, you're going to have healthy volunteers. The first part of the focus of phase 1 is not whether it does what it's supposed to do, but is it safe? Does it work? Side effects? Are there any dose or side effects? We're going to look at what would be the minimum effective dose and the maximum effective dose. Okay? And if that looks good, you go to phase 2. Phase 2, you have 100 to 200 healthy volunteers. There's something you got to keep in mind, too. If it is a crisis and if there are no other treatments out there, no other alternatives or anything else, sometimes phase 1 and 2 are fused together. Particularly, this is for situations where the disease is lethal and we have no other means of treating it. But let's keep phase 1 and 2 separate. Phase 2, what are the most common side effects? And also, how does the immune system respond? Now, this is a very important point. When you look at a vaccine, it has to provide 2 forms of defense, which relate to the adaptive immune system. It has got to have a humoral response, and that means B cells. B cells have got to be able to make antibodies to the particular antigen that you're putting in as the vaccine. You've got to also have your other, besides the humoral, you're going to have basically the cytotoxic effect. B cells will release antibodies. T cells go after cells that are already infected. See, antibodies can't get inside of a cell. So if the cell has been infected by a virus, let's say, you really can't go after them with B cells. What you can do is have T cells that will begin to take action. They will be able to detect certain markers on the exterior of that infected cell, and they will release certain substances that eventually kill that cell and thereby stop the replication of the virus. Now, phase 3, hundreds or thousands of healthy volunteers are present. They compare basically the vaccinated to the unvaccinated. Again, keeping in mind, is it safe? Is it effective? Are there common side effects? And this is where they try to find, okay, 4 out of 10 people, or 4 out of 100, or 5 out of 1,000 will have an adverse effect. And depending on the adverse effect, that may be go or no go. But then what happens? Now, all of this is on the manufacturer. They pay for this. And just to help you to understand it, the manufacturer, once they have gotten the patent and they're in the lab, we think we have made this new. A patent only lasts 20 years. Okay? The latest studies have been that to get a new drug or vaccine out into the market cost you $628 million and takes about up to about 10 years to get approved. Okay? Now, with that going on, sometimes they've had a few drugs that went through phase 3 trials and failed. There goes all the money, there goes a bunch of the staff laid off, etc. Now, meanwhile, as they're getting prepared for this, the manufacturer will continually test the vaccines themselves and allow the FDA to inspect the facility, and that's the facility making the vaccine, for quality and safety. They will continue to monitor outside the manufacturing process as well. So the CDC and the FDA sponsored surveillance program, which is the Vaccine Adverse Event Reporting System, VAERS, will be present. Usually what happens is this: once you get done with the phase 3, you bring all this lots and lots of really cool data to the FDA. The FDA looks at any benefit risk, any safe, effective, etc. Now, the FDA has a committee. We're talking about a bunch of doctors, immunologists, specialists. Sometimes they're going to bring in some people from the ACIP. They will review, have experts that will review the clinical trials, etc. Monitoring ongoing, even after recommendations are made. CDC director may get involved, and they review recommendations, and the official policy is published in CDC's Mortality and Morbidity Weekly Review document. It's kind of like a small book. Okay. How do we monitor after it's hit the market? This is where we get into the Vaccine Adverse Event Reporting Systems, VAERS. Now, this is a passive surveillance system. It will collect and analyze adverse effects, side effects, okay? And you really do have to have that sometimes. The manufacturer will continue to test all the lots to ensure safety and purity. So they have their internal testing going on. The FDA monitors and inspects the facility, so they want to make sure that it's clean and proper, and every batch is basically the same. But then we get into what happens if, somebody takes it, gets the vaccination, and has a heart attack later on. Well, they will report this to VAERS, and then from there will be an investigation. Sometimes vaccines have had to be pulled away. There was a vaccine for rotavirus for little kids, for infants, and it was pulled away. And the reason being that it caused gastrointestinal blockages for some unusual reason. There have been other vaccines that have been shifted. So instead of being attenuated, they are now killed viruses, such as with polio. In some places in the world, they're still battling polio, I might add, basically because of culture. In the United States, we pretty much killed off polio. Okay? I think I included it, but if not, you can look in YouTube. There's one video of one man. He's the last person on an iron lung. And to help you what that is, when polio goes up, it begins to paralyze all the motor neurons, etc. And so these individuals, to survive, just to keep breathing, are put into this large cylindrical device. And they have a mirror where they can look up and talk to people and everything, but basically they can't move their arms or anything else. They're alive, they're alert, and it just happened that they got it, and it was severe enough. And here's the sad thing. Now, this one individual is one of the last ones to have a iron lung. The iron lung machine keeps them alive, but there's no more manufacturers that make iron lungs. As a matter of fact, he has to get basically a skilled tradesman to make replacement parts and things like that. How about that? Okay. And this is just a couple of other pointers here. I mentioned about passive surveillance. This is the systematic surveillance of the health outcomes or exposures, in contrast to systems which providers and laboratories are contacted. Passive surveillance relies on submissions by the wider public. Then we have to deal with one thing that I'm sure many of you have seen in the past several years when it came to the COVID vaccine. Vaccine hesitancy. This is the reluctance or refusal to vaccinate despite the availability of the services. Now, I know that that's a mixed bag for a lot of people when it comes to dealing with the COVID vaccine. But we unfortunately still have people that have vaccine hesitancy for their own children for measles, mumps, rubella, diphtheria, tetanus vaccines. And sometimes that leads to some serious health problems down the road. And as I mentioned to you before, there was a question about how vaccines work. Keep in mind that vaccines are a type of modification of the adaptive immune system. Now, what I mean by that is that you do not have to get the disease. You get the antigens as part of the vaccine. And so the primary cellular immune responses are in sort of this pattern. First, you have macrophages. They're going to engulf the pathogen. In this case, it's not a pathogen that they're going to engulf. They're going to engulf the components of the vaccine. B cells will then use the antigens that are released to generate antibodies as plasma cells and as B memory cells to remember prior exposures. Remember that a B lymphocyte will not only be the cell that releases antibodies and fights the good fight, but some of them are going to be held aside, just like reserves, and be circulating in case they ever come up in front of the disease again. That's why they're called memory cells. T cells have the same thing. Okay? Now, these cells are going to be primed to respond to specific antigens, both the memory cells. Killer T cells are a specific type of lymphocyte. They also recognize a foreign antigen, but in infected cells. Again, how do you get inside the cell? They can't get through the cell membrane. What happens is the cells respond by sending off signals or whatever, and so you have a situation where the T cells will detect this. They will release certain materials. They're called the grouping cytokines, and they will act to either destroy the cell, trigger apoptosis, which is programmed cell death. They may release certain proteins that kind of form holes to punch holes into that cell and thereby kill it, etc. Okay? And so we're at the end here of week 3. First, review the lecture. Review the lecture PowerPoints. Yes, I will correct the spelling errors. Read all the supplemental articles in Module 3. This will give you a good handle on a lot of different things. View all the videos in Module 3. Participate and complete the discussions for week 3. Oh, yes, and some of the discussion assignments are going to require you to review this week's videos or supplemental articles. You're expected to do research in this class. Make sure you cite it. Complete the quiz for week 3. One more thing about the quizzes and the exams. They will be including the lecture PowerPoints, the supplemental articles, the chapter readings, as well as the videos. So you want to stay on top of all of that. Have a great day.